Good afternoon, students.
Let's start now.
Yes. Yeah, please start.
Good afternoon to one and all present. We, Darsh Desai, are delighted to welcome you all to the IIC session of the 5th Student Research Congress, organized by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by University of Mumbai, along with our esteemed industry partners, ACG Worldwide and SciTech Center. Dr. Raghunath Mashelkar, an eminent Indian scientist, once remarked, innovation is not a luxury, it is a necessity. It is the art of creating affordable and sustainable solutions to address the greatest challenges of our time.
The Innovative Idea Challenge presents an exciting platform where creativity meets practical application. Participants are encouraged to think outside the box and develop ideas that not only showcase originality but also hold the potential to drive real world change. This challenge fosters an environment where science and innovation converge, allowing participants to explore and implement solutions that address complex problems with a fresh perspective.
At its core, the challenge celebrates the importance of visionary thinking, ideas that challenge existing norms and inspire progress. By combining inventive approaches with scientific methodology, participants are equipped to craft solutions that are both imaginative and grounded in feasibility. The event encourages collaboration, experimentation, and a commitment to pushing the limits of what's possible.
As we come together in this space, we honor the relentless drive of innovators who strive to create a meaningful impact. The pursuit of such groundbreaking ideas is a powerful force that can lead to transformative changes in industries, communities, and beyond. The Innovative Idea Challenge is a testament to the belief that with the right mix of creativity, knowledge, and determination, ideas can spark lasting changes in the world.
I extend a warm welcome to our esteemed judges whose knowledge and perspectives will undoubtedly encourage and guide our participants towards excellence in innovation. I'm privileged to welcome the distinguished judges for this event, whose expertise and insights will inspire and guide us all.
The judges for the IIC category are Mr. Vijay Shivpujay, Dr. Srinivas Savle, and Dr. Meenal Rana.
Can I take pitch in a little bit? Thank you very much,
Yes, ma'am.
Dr.
Please.
Vijay and Dr. Savle and Dr. Minal for accepting our invitation to be the judge for this session. And I must share my thoughts here that this is the IIC Institute. It's an innovation challenge entries, and who else other than the CEO of Atal Incubation and the patent attorney and the faculty who has good number of patents, the right combination for the judges. So thank you very much, and I'm sure the participants would be delighted to have your insights in form of questions, and that will also pave the different road to think further. Your insightful discussion or the questions in form of questions will open up a new window for them to think. So sometimes it is a thinking out of the box or it is thinking with a different lens. I'm sure three of you will have a major impact on a student's journey of innovation. So thank you very much. I really appreciate your time and efforts today. Thank you very much.
I would now like to introduce Mr. Vijay Kumar Shivpuje. He is a multifaceted professional with an impressive career spanning over 19 years in the field of intellectual property rights, pharmaceuticals, legal consultancy, and knowledge process outsourcing. He is the director of Patlex Business Solutions, a premier firm specializing in patents and designs, and the CEO of EduLiterati, an innovative online education platform fostering skill-based learning through collaboration with leading universities and institutions. Sir holds a master's in pharmacy, an MBA, and an LLB, and is a registered patent agent with the Indian Patent Office. He has actively contributed to India's entrepreneurial and innovation ecosystem, serving as an IPR advisor to SOCAM, a mentor of change for Niti Aayog's Atal Tinkering Labs, and a mentor with Startup India under the Ministry of Commerce and Industry.
Renowned for his expertise in IPR, Vijay Sir has conducted over 300 awareness programs across universities, colleges, bar associations, incubation centers, and industry chambers. His exceptional contributions have earned him several accolades, including Asokam's Award for Outstanding Contribution in IPR Awareness in 2021 and the prestigious IP Mentor of the Year Award in 2024.
A visionary leader and educator, Sir is committed to fostering innovation and skill development, empowering individuals and organizations to thrive in a knowledge driven economy. Thank you for honoring us with a valuable time and being a part of this occasion.
Our second judge for today is Dr. Srinivas Savle. Dr. Savle is a distinguished leader in the pharmaceutical and healthcare sectors with over 23 years of experience spanning drug research, development, regulatory compliance, and the innovation ecosystem. As the CEO of AIC LMCP Foundation, an Atal Incubation Center hosted by the LM College of Pharmacy in Ahmedabad and supported by the Niti Aayog, he spearheads initiatives in pharmaceutical and healthcare innovation.
Dr. Savle holds a PhD in pharmaceutical sciences from Gujarat University and has a prolific career, including leadership roles at Torrent Pharmaceuticals, Syngene International, and the Torrent Research Center. He specializes in regulated bioanalysis, biopharmaceutics, early clinical development, GXP compliance, electronic data workflows, and vendor qualification, among others, supporting CROs and pharmaceutical organizations in achieving excellence in these domains.
Actively involved in academia and the innovation ecosystem, he has contributed as an adjunct professor, mentor, and committee member at LMCP. His professional affiliations are extensive, including leadership roles in the Indian Pharmaceutical Association, Gujarat branch, the Society for Pharmaceutical Dissolution Science, and international organizations such as the AAPS and Global Bioanalysis Consortium.
Dr. Savle's contributions have been recognized with prestigious awards, including the BioContribute Award and Best Research Paper Award by the IPA. A prolific scholar, he has authored several publications and book chapters and has been an invited speaker at numerous national and international conferences. His steadfast dedication to advancing pharmaceutical sciences and fostering innovation has made him a prominent figure in the global healthcare ecosystem. We deeply appreciate your time and presence, which have created a great value to this event.
I would now like to introduce our third judge for the day, Dr. Meenal Rane. Dr. Meenal Rane is an accomplished academic, faculty, and researcher serving as an assistant professor in the Department of Pharmaceutics at SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Mumbai. With a robust educational background, she holds a PhD from SMDT University. Dr. Rane brings over 11 years of academic expertise, complemented by 3.5 years in the pharmaceutical industry, including roles at SciTech Center, Caves Private Limited, Prana Hyperbaric Oxygen Center, and Marico Industries.
A prolific contributor to pharmaceutical research, Dr. Rane has 11 publications, 2 granted patents, and 2 published patents, with her work cited 51 times. She has successfully secured industry and government grants exceeding rupees 1 crore, fostering innovation and collaborative research in the pharmaceutical domain. Dr. Rane has mentored 20 MPharm students and has actively contributed to academic excellence through more than 50 research presentations, conferences, workshops, and seminars. Her dedication to advancing pharmaceutical education and research is further reflected in her role as an invited speaker at various prestigious forums.
All of our judges, thank you for generously dedicating your time to the excellent.
Thank you, students.
Before we begin, I'd request all the participants to make note of the instructions carefully. The presenters have to rename themselves as per the allotted codes. The presenters are requested to stay online, switch on their camera when their code is called out. Pre-recorded video will be presented on the screen. Participants cannot reveal their names or any affiliations. Each participant is requested to stay alert for their roll call. All the participants should be present for the session. All members from the group are allowed to answer during the question and answer session. The duration for the question and answer session is limited to 5 minutes.
With this note and the permission of our respected judges, let's begin with our oral presentation competition as all of our participants and audience are eagerly waiting to present and learn about the new, unique, and interesting ideas that will be presented here. Also, I kindly request the judges to confirm that they have received the scorecard. I would, I would
Yeah,
now
I have
like
received.
Thank you, sir.
Thank you. I would now like to call upon code number IIC03 to be ready. Soon we will be presenting your video. Please switch on your camera.
Good afternoon to one and all present here. As per to the recent reports of India Today, the proportion of world's population over 60 years will nearly double from 12% to 22% by 2030. 1 in 5 will be senior citizen by 2050. The increasing expectancy and leading fulfilled life of youth ensuring the quality life of the elderly individuals become essential for social and economic sustainability. As we progress in our lives, the elder population do feel left out and build a fear of loneliness among themselves. A major issue among the geriatric population is regarding their mental health. In some senior citizens, due to physical limitation or lack of accessibility, they have to compromise their independency of life. Huge problems faced by geriatric populations are lack of knowledge of therapy and medications. Elder people mostly fail to manage their medications and appointments, pathology tests, and so on. A companion is needed. When needed, a proper attention is required to maintain the proper time of medicines and help scheduling daily checkups, and this make it a health or record of the individual. They usually go through a social isolations. Around 1 in 4 individuals face social isolations problem after losing their loved ones. Social isolation can be further lead to depression. According to recent studies and surveys, around 43% of senior citizens have been reported feeling lonely. The vision. The ideal vision to create a device which would promote independence to elderly individuals, enhancing their quality of life. An idea of inventing a device for geriatric population can be a solution to above-mentioned problems. A device that can take care of geriatric citizens when needed, the healthcare access who can solve issues like loneliness amongst the elder individuals and the problem of mismanagement of the medications can be resolved through a device. Presenting a device named Sakha, who could be an excellent companion when needed. Sakha, meaning a friend, who will support and take care of senior population. A useful mergence of an AI and mechanics can result into a boon for elder population. A wearable device that would be convenient to wear, which will function as a great associate to an individual by installing AI techniques such as ChatGPT to chit-chat with the individual when required. Sakha would be a hanging device like an identity card for the convenience of the individual. This would promote more independence as it will easy to use with less amount of help from the another individual. The features. Sakha can engage in meaningful conversations and would be a great advisor. The person can freely share his or her emotions to his Sakha and could feel more listened and can express more rather than keeping things within the mind, which can lead to depression and anxiety. This would provide emotional support. Sensors can be used to alarms to the user about the medicines and appointments. Timely reminders can be a solution of accurate intake of medicines. Tracking of vitals like heart rate, oxygen, using sensor can be indulged in the device to record the vitals of health and help to maintain the health records. The function in the device like playing favorite music, reading out books, giving news updates, weather information can be built using AI features. Technologies used in this device are like alarm system for timely reminders, sensor system, sensor system for monitoring heart rate, pulse rate, oxygen rate to maintain the record of activity of the personal individual. AI system for interactive conversation and communication, and mechanical system obviously to make a device. Sakha would not only reduce the emotional baggage of loneliness but will empower the elderly population by enhancing their independence. It will also enhance and fulfill their life while by talking and the communication and the interaction interactive conversation. It will give a proper attention on the medication. It will provide timely reminders, also will be helpful to manage and schedule appointments. From the patients in the hospital to elders in the care centers, they all need one thing, that is support. And that is what Sakha is for. As sad it is not everyone can live with their loved ones, be it due to geographical distance or any other factor. Sakha doesn't mind filling the gap. Sakha will not just be a device, it will more likely to be as friend, a guide, and a support system. Let us use the technology to build just as smarter devices, but also stronger connections. So these are the references which are used, and thank you.
I now invite the judges to ask their question to the respective representatives. But first, I request IIC 03 to please turn on their camera.
Uh, yeah. So the first question is, have you actually prepared this device, or this is just a concept which is in making actually?
Uh, Good afternoon, sir. So
Yeah.
this is just a concept to present to the geriatric patients or population. This is not, I have not prepared yet, but this is the idea. So I shall look up to it and it can be invented.
How did this idea actually turn up, or when did you start thinking, and was there any trigger that you thought in this specific direction to come up with this kind of medi companion?
Yes, sir. I've actually seen in my family kind of the older people doesn't get much attention. They are like the attention is needed to them, but as like the schedules are busy of ours and everyone, but looking up to them is very necessary in my opinion. So building this can be a solution to them as they may not feel lonely and usually people feel lonely, the stage of the life where they have to speak very much, like they have to speak a lot, but they don't have someone to listen. So Sakha could be the one who can listen. So the idea came by this. That's it, sir.
Yeah,
I
done.
request code IIC03 to please turn on your camera.
Yeah, Minal ma'am and Srinivas sir can take it ahead. I mean, if they have any other questions.
Yeah, I just had a kind of follow-on question. As this is so, have you thought of like the features you have listed? One as a listener, one as reminders. You also have some content for listening to them, right? And you also talked about monitoring the vitals, right? So, any thoughts on, because you're trying to address a variety of uh, issues with the single device. So, how is it? Is it a you are conceptualizing this as a hardware software combination offering, or it's only a software based solution? What is that is in your mind? You are on mute. Can you unmute yourself?
Yeah, so, sorry, sir. Actually, it is the AI and mechanic mergence, like a software and a hardware mergence, where, as we can see in the smartwatches, we can sense our vitals with the sensors, the laser ones. So we can build in that device, which can be sensed on the wrist or for the heart rate and pulse rate, and it can be further like for the body temperature and everything. But I'll look up to it. I'll take a note of that. The temperature one, I haven't thought about that, but the sensors, as in the smartwatches, can really be helpful for monitoring the health vitals.
Quickly, have you just looked around? Have you searched about anyone else is working on similar solution?
No, sir, not yet. But I'll see. I'll surely see. I
Means
haven't seen it.
if at all this idea is to be taken forward, you also look at well the possible competitors.
Yes, sir. I'll look up to it.
Yeah, I'm done.
Okay. So I have a question for you. Before you thought of this, had you done any survey to come to this problem statement? What was the need for thinking on this concept? Did you do any persona sketch or any interviews with the geriatric people? Are they really looking for this kind of device?
No, ma'am, I haven't used any research or interviews. I haven't done it. But I think like they do need, because many like geriatric population, they usually feel alone. We can sense that. They have to talk a lot, but they have no one to listen. So it is my perspective. I haven't done any research, but I'll take this idea, I'll surely do that interviews and research for building this idea more significantly.
Hello, am I audible?
Yes, ma'am.
Hello, am I audible?
Yes, ma'am, you're
I think there's
audible.
some network issue. Yeah, so I hope you could hear my question, or you need to repeat it.
Ma'am, I answered your question, I guess.
So I think there was a network issue. Could you please repeat?
Okay, so I haven't done any research or interviews about regarding this, but I surely think it is one of the like a concept that must be given attention to. I feel that they need to be listened, a stage where they have to talk a lot but there is no one to listen. So according to me, this must be the idea that can come up with the solution. And if I further take this idea to make like a real one, so I'll surely do the research and interviews accordingly.
Thank you from my side.
Thank you, judges.
I'm done.
Now, I would kindly request all the participants to note that turning on your camera is mandatory, or else you will be disqualified. Please make a note of it. Now, let's continue with our session. I would request code number IIC04 to be ready. We will be presenting your video shortly. Thank you.
Think about Priya, a busy working mom managing her health while caring for her diabetic father. She often forgets to give him his evening medication. Or think about Ramesh, living in a remote village, unsure how to store or dispose of expired antibiotics. Stories like theirs is what inspired MedEasy. MedEasy is a personalized medication interaction advisor app designed to make medication management efficient. Every year, 1 in 10 patients globally is affected by medication errors. These errors can lead to severe health complications, prolonged recovery, and even fatalities. Managing medication, especially for those on complex regimens, can be overwhelming. That's why I created MedEasy, a personal medication interaction advisor app designed to make medication management safer, simpler, and more efficient. MedEasy is for anyone who wants to manage their health better, but it's especially helpful for geriatric patients, fitness enthusiasts, patients with allergies or sensitivities, patients managing multiple medicines and supplements, patients who are going through rehabilitation, parents of young children to manage pediatric prescriptions, and rural and remote area patients. So, how the app functions. MedEasy simplifies medication management with an intuitive and user-friendly approach. Users can input details about their medication, dietary habits, and prescription, enabling the app to provide tailored usage guidelines, warnings about potential side effects or interactions, and tips for proper storage to maintain its potency. With a built-in barcode scanner, users can easily register medications by scanning their barcodes, allowing the app to track expiry dates and send timely reminders for replacements. To ensure users never miss a dose, MedEasy sends alerts specifying what medication to take when and in what dosage. Additionally, the app promotes eco-friendly practices by educating users on proper disposal methods for expired or unused medications, helping to reduce environmental pollution. Medication management often presents several challenges, but MedEasy offers effective solutions to tackle them. Missed doses are a common problem, and MedEasy's personalized reminders help users to stay on schedule. The app also mitigates the risk of harmful drug interactions by providing warning-based input for users. For expired medications, MedEasy uses barcode scanning and expiry alerts to ensure users always have safe and effective medicines. Improper storage, which can reduce a medicine's potency, is addressed with detailed guidelines to preserve its efficacy. To simplify confusing medication labels, MedEasy offers easy to understand explanation, minimizing the risk of misuse. Furthermore, the app supports eco-friendly disposal practices, helping users reduce pharmaceutical waste and its environmental impact. So, why does this stand out from the other apps available in the market? Among a crowded market of pharmacy apps, MedEasy distinguishes itself through its innovative and user-focused features. It goes beyond basic refills and reminders by offering personalized guidance to tailor to the user's dietary habits and specific needs. The barcode scanning feature not only tracks expiration dates but also provides storage tips ensuring medications remain safe and effective. Unlike most apps, MedEasy promotes eco-consciousness by educating users on responsible waste disposal by combining interaction warnings, storage tips, expiration alerts, and waste disposal education into a single platform. MedEasy provides a holistic approach to medication management, eliminating the need for multiple apps. So, in a world where medication errors impact millions, MedEasy is here to change the game. It's not an app, it's a companion that empowers users to take control of their health while promoting sustainability. So, let's move forward to a future of safe medication practices, healthier lives, and a cleaner planet. Thank you.
I now invite the judges to ask their question to the respective presenters.
Yeah, I'll start with a simple question of how is the data entered into what have you thought of? How would the data be entered? Would the patients be entering the data in the app, whatever medication they have got?
So the patients can enter the data manually, or they can scan the barcodes, which would make it more convenient. And after they do input the data, the app will confirm it. Like basically, they will confirm if the medication that they have entered in is the medication, because often patients can make errors while inputting them like manually or like.
Yeah, and that may be a biggest constraint actually if the patient has to enter the data, maybe, and when you think of especially the rural areas, that point of time, it would be a big challenge as such. When the patient himself or herself has to enter the data, because the entire system, the app system is dependent on the data entered by the patients as such. And that's what I felt is something which may be a challenging actually.
I mean, the app will obviously double check the medication that is input by asking the user if that is the medication. Otherwise, the barcode scanning obviously exists, and they can just barcode scan it, and the data will be input into the app.
Okay, honestly, I couldn't understand the context of putting it from the patient's end actually. Oh, that's what I have. Maybe Dr. Minal, you can take it ahead. You're on mute.
Yeah, so I have a question similar like sir, that how will the patient add the data and up to what extent, how much time it will take for patient to enter the data? If it is going to be for a multiple medications, have you thought on this?
Yeah,
If you have a patient who is on multiple complications.
Yeah, so like I said, the app is very user friendly, right? So they can either input the data by themselves, or usually medicines have barcodes on them, right? They can just scan the barcode and the data will be like input into the app. So they don't have to do much hassle of entering it manually.
So what data are you expecting? What data needs to be entered? Can you give me an example? What information has to be read?
Just the medication names, because the app will like provide the doses for it, and like the reminders to take the medicine. So honestly, just the medication names, and the app will provide how much to take it. They can also scan the prescriptions, so the data will directly be input into the app at one go.
So
in fact, this is exactly,
how can we?
Yeah, this is exactly what I was expecting. Yes.
How can we validate the name of the medicine which has been entered is having a correct spelling? One small error can lead to wrong information fed to the device, right? So how will you validate this point? Have you thought on this?
Right. So, like I said, the app will double check it. It will again ask you if the medication that you have input is the medication that you need. So there is a double
What if
check
the patient and what if patient is not still able to give the right spelling? If I don't know the spelling, how am I going to correct it? I'm to say I feel that's a constraint. It will be more restricted to only people who are educated. Your educated people also can't spell properly.
exists where it can convert speech to text.
and they can input their data in like that as well. But otherwise, the barcode scanning feature is already there, and so is the uploading the prescriptions directly. So if if the patients don't want to do much of a hassle by putting the data in, like manually typing it themselves in, they can always barcode scan it or enter, like take a picture of the prescription and upload it onto the app.
And is there any possibility of you having this data input into different language? Is it possible?
Yeah, obviously, there will be like multi languages because again, there are people who speak different languages or they cannot understand a specific language. So it will be available in multi languages. And also for the people who cannot read well, again, the voice input is already there, which will convert speech into text.
My last question: have you actually demonstrated this, or this is an ideation pitch?
This is just an idea. I have not really demonstrated it or made the app.
Okay. Oh, thank you.
All right,
Thank
thank
you,
you.
sir. Srinivas, you can
If
take
I
it.
may ask a couple questions, yeah. I think uh, so. I have few questions. So when you are working on this idea, who all are there in your team?
It's just me.
Just you. So what is that you can do yourself
Sorry,
in this?
can you repeat the question?
If I have to execute this, if I have to execute this solution, what is that you can do on your own?
Like create the app with
Yes.
the idea that I have came up with.
So do you have skills to build the app?
Not really, but I can involve other people.
Yes. So
To make.
what are those skill set you will need to start working on this idea?
Well, to start off, I will need people to create the app for me and like code the app properly so that it works seamlessly. And along with that, I'll also need like a marketing team to like um, make my app known to people that an app like this exists and it can help.
Yeah. So can you visualize what all kind of data you need to build in? Like simple statement you made that the app will verify the input from the patient. So
So
do you know what goes behind? What needs to be done for uh, kind of evaluating whether the information is correct?
like you're asking like the features of the app.
No. The statement you said, right? The app will verify
Double. Yeah.
the information, double check. That's the word you used. So what do you need to build for that? What goes behind at the back end?
I'm sorry, I'm not aware of that.
Yeah, so how can an app double check the information?
Like usually they can verify, right? So for example, if I need to take medicine and I input that, it will flash an alert on the screen giving the name of the medication that I just input with the statement saying like, is this the prescribed medicine or are you sure this is the prescribed medicine? To which the patients can accept or like deny the alert that just popped up on the screen.
So there needs to be a lot of data that is already built in to cross
Yeah.
check.
Yeah, there needs to be technical
Yeah.
work done.
And secondly, you're trying to build some other features which are not just linked to the information of the medicine. Talking about, so you need to, for addressing each feature, you have four major features. Each feature needs a huge database to be built in if you want to seamlessly work as a complete solution. I think there's a lot to think through. No doubt it's an idea, but like if you really want to work on it, you basically see whosoever wants to build a solution, you bring in pharmacy insights, the pharmacy knowledge, but you lack for the technological understanding of the IT component of it, right? So my just one observation, I couldn't ask for the earlier presenter, but are you presenting as a team or you're like a solo presenters for your ideas?
I'm a solo presenter and
Okay,
it just
thank you.
all right. Thank you.
Thank you, judges, for a valuable question. Let's proceed. I request code number IIC05. Kindly be ready. We'll be presenting your video soon. And please turn on your camera.
Imagine a future where the clothes you wear are not just a reflection of your style, but a tool to improve your health and well being. This isn't just a dream, it's the reality we're stepping into with Medicator, where healthcare innovations meet cutting edge technology and design. This is more than a trend, it's a revolution. Here's why it matters. Today, over 1.8 billion people suffer from severe skin conditions such as eczema or epidermolysis bullosa. Severe skin disorders are more than surface level issues. They can be life threatening. Conditions like melanoma claims lives every year, while untreated infections and chronic skin conditions often lead to systemic complications. Psychological disorders, often called silent killers, affect millions of people worldwide. Depression alone impacts over 280 million individuals, while conditions like schizophrenia, bipolar disorder, and PTSD disrupt countless lives. Substance use disorders, SUDs, disproportionately affect underprivileged areas, where limited access to healthcare, education, and economic opportunity exacerbates the problem. The World Health Organization estimates that over 35 million people worldwide suffer from SUDs, yet only 1 in 7 receives the treatment. Traditional treatment methods for chronic and psychological conditions often involve a combination of oral medications, injections, and therapy sessions. While effective in some cases, these approaches have several drawbacks. Medications can cause side effects, require strict adherence to dosing schedules, and may be inaccessible due to high cost. Therapy sessions demand time, commitment, and resources that not everyone can afford. In conditions like eczema or schizophrenia, treatments can be long term or even lifelong, further amplifying the burden on the patient. An upgraded medicine and treatment procedure for severe skin, psychological, and substance use disorder is not just necessary, it is critical for several compelling reasons. Current treatment for severe skin, psychological, and substance use disorder often provide only partial relief, failing to address the root cause. These conditions are becoming more prevalent globally, increasing the burden on individuals and healthcare systems due to the prolonged, costly, and inefficient treatments. As these disorders can be life threatening, there is a pressing need for quicker, more affordable solutions. Advances in personalized medicines, bioactive therapies, and digital health technologies offer new opportunities for more effective and precise treatments. The stakes are high, and the need for innovation is critical to meet this global challenge. So, what's the solution? This
I'm sorry for the inconvenience. There's something technical glitch here.
in biotextiles and microencapsulation.
Sorry for the inconvenience. Let's resume now.
At the intersection of healthcare technology and microbial scrubs can provide an additional layer to protection against the infection. Weighted blankets infused with calming agents can help those with PTSD or anxiety disorders. These examples shows how Medicator's applications are vast and adaptable to specific health needs. The core of Medicator lies in microencapsulation technology. This involves embedding microscopic capsules containing therapeutic agents into the fabric. Let's break down this on how it works in depth. First, find a suitable textile. Second, choose your active agent concerning your disorder. Third, creation of microcapsules. Therapeutic agents such as drugs or nutrients are enclosed in microscopic capsules made of biocompatible materials. These capsules act as tiny protective shells, safeguarding the active ingredients from the degradation. Now, integration of these microcapsules into the textiles. The microcapsules are embedded into the fabrics of the fibers using advanced technologies and techniques like padding, coating, or spray application. These method ensures that the capsules are securely attached to the material without facing or affecting the garment's texture or appearance. The fifth is testing and quality control, and the last is final product design as per the requirements. So, how does the penetration really work? It works by the trigger mechanisms. Microencapsules are designed to release their contents in response to specific triggers: friction, body heat, moisture, or sweat. Delivery to the skin. Once released, the therapeutic agents are absorbed through the skin, our body's largest organ, using one or two pathways: localized effect and systemic effect. Sustained release. The microcapsules can be engineered for controlled gradual release over time, ensuring a steady supply of treatment and reducing the need for frequent reapplication. The continuous controlled delivery for therapeutic agents offer non-invasive, personalized treatments that enhance medication adherence, reducing side effects, and improve patient safety and comfort. Targeting medication directly to the skin maximizes effectiveness while minimizing the waste. This approach improves quality of life, simplifies caregiver support, and is adaptable to daily routines. It also promotes sustainability, ease of use in healthcare settings, and real-time monitoring to ensure safety and timely adjustments, contributing to more efficient and eco-friendly healthcare. Fast fashion is a $106 billion industry generating 92 million tons of waste annually and consuming 79 trillion liters of water per year. Medicator offers a sustainable alternative by repurposing defective, discarded, or overproduced garments into biotextiles. These reduces waste while creating life-saving solutions. By marrying sustainability with innovation, Medicator addresses two global challenges: healthcare accessibility and environmental degradation in a single stroke. The Medicator business model focuses on the production and sale of bioactive textiles for the healthcare sector, targeting both B2B, hospitals and government sectors, and B2C, patients and private sectors market. With an estimated manufacturing cost of 250 rupees per garment, the model operates on a dual pricing strategy. This approach ensures higher profitability into the B2C market while maintaining sustainable revenue from bulk sales into the B2B market, creating a scalable and financially robust business model. Medicator is more than a technological breakthrough. It's a movement. It envisions a future where healthcare isn't confined to hospitals, but integrated seamlessly into daily life. Imagine a world where your clothes takes care of you, adapting to your needs and making treatments effortless. This vision is driven by a generation, our generation, that values innovation, sustainability, and inclusivity. Now it is the time to take action. Whether you're advocating for sustainable practices, inventing in wearable tech, or simply raising awareness, you have the power to be a part of this transformation. Let's not just imagine the future, let's wear it. Let's create a world where health, style, and technological processes walk hand in hand. Together, we can make Medicator the next big leap in healthcare. Because when style meets cure, the possibilities are infinite. Thank you so much. Thank you. I now ask the judges to for their questions. Kindly switch on your camera. IIC 005. I'm so sorry, but I can see my camera on the screen. I don't know what's the issue with me not getting onto the spotlight. Try to switch it off and then turn it on again. How about now? Can you see me? No, but wait, I'll check with my team and yeah, sure. Okay, we'll continue with our questions. Judges, please ask your questions.
I
So
request the
may I ask the
judges to
question?
please complete the
Yeah,
question.
yeah. Okay, the team, can you mention any one drug which can be actually like used it with this particular kind of platform as such? Honestly, I couldn't uh, get much actually as far as the platform is concerned. When you mentioned that clothing with the medicine, how is it controlled basically? Because the be it a drug delivery or the prescription, it is controlled, it's regulated basically, right? So can you let me know any one drug just to understand how it would be actually working?
Yeah, sure. As this topic is a really early research of topic, so I should start it from a really initial stage. So this is basically idea which is taken from the space aerospace companies. The space suits basically use micro encapsulation technology in the early 1980s to manage the thermal buffers. So that is where the thermality, or what do you call it, is the actual managing of the drug really works. So one drug that can be encapsulated that I've also mentioned in my PPT as well, which is for the skin disorder, which is pemphigus. So we can embedded corticosteroids. The corticosteroids are the current treatment which are used for the cure of the disease. So, bioactive textiles and microencapsulation technology prevent an innovative and non invasive solution to manage this. And we can use corticosteroids as a core in microencapsulated capsules, which manages the drug delivery and its degradation from any sort of workings. And how does this penetrate is basically with the trigger mechanism, the sweat and the moisture. It penetrates to that, and it's the same dermal, transmal dermal delivery that we see in semi-solid dosage forms as well. And it's the same working.
How have you calculated the final figures that you mentioned, 50 and 40% profit margins actually? How have you evaluated that?
Well, it's just an estimation. Well, a cloth with microencapsulation technology wouldn't be as a selling price of 250 rupees or 300 rupees because this technology is really expensive. It's just a round figure to show people that, oh, this is how it should work. But at the end, it's a technology that would be open to the subsidized program because it's at the really initial stage. we really need to research on this topic and work on how the procedure would cost, how the manual major people that working onto this project will cost them, what will be their job profiles. That is what comes up with the end of the money plan. But this is just an estimation of how it might look to B2B and B2C business programs.
Okay, thank you. That's it from my end. Yeah.
Thank you so much.
Okay, so I have a question for you. Uh, how have you arrived to go for this concept? What was your homework for it? I could see.
Okay. So, to give like a really fancy answer to that, or maybe just be honest to it, for me, I think when I think of a problem, I ask myself what is a big problem for me, so that if I solve it for myself, I would be able to do it for other people as well. For me, my biggest thing is skincare. Like sunscreen is such an important thing now these days, and I hate reapplying it after and after over and over again. So we have this thing in China and Taiwan, which is called as UV protective jackets, which is something a lot similar to sunscreens because it helps you from the UV protector. And as I mentioned in the PPT as well, you can save yourself from melanoma, which is a skin cancer. So I went on researching on this topic and I thought, how easy and comforting would it be that your clothes actually work for you? Because it sounds impractical at first, and someone thinks of it like how a drug would penetrate from clothes. But these are some things that actually has been established when it comes to different space suits. So you have fragrance textiles as well, like luxurious brands like Hermes. They use microencapsulation technologies to create silk scarves and ties. So why not something basic, and why just use this thing into a drug embedded technology? Like that could be a big thing. Like if you think about innovation, this is what I come up with. It's more
Okay, so
comforting, I
it's
think.
your own perception. So it's built up on your own perception, right?
Well,
I can say
it
that.
is, but at the end, if I'm focusing on hospitals, you have a lot of coma patients. You have people who have been suffering from really lifelong skin disorders, and these are really painful procedures. These are costly as well. So why we make something that lasts you long for 1 to 6 months and also reloadable, and it costs you less, and there isn't much of drug remembering, as you can say, oh, this is something that I need to apply every second or every hour. It's just a comfort of how a dosage form can be comforting and also be more cost effective to people.
Okay. Okay. So now coming to the cost, um, can you just estimate what would be the cost comparison between a product to be applied for melanoma and a jacket to be applied or to be worn for melanoma? How will be the estimation? Have you estimated the cost if somebody has melanoma?
Yeah.
What will be the cost of the jacket?
Well, it really depends on what kind of technologies are we working on to. For me personally, it is a business plan. I work alone in this team, and I do need people who expertise in every single problem, because for me, cancer is a different thing, skin disorder is a different thing, and substance use is a different thing. You can't use the same drugs all over again. Melanoma is again a thing which can be the cure can be through surgical excisions. You have immunotherapy, you have chemotherapy, but enlisting those things into a jacket is again a different field of research that I'll be working on to understand what drugs are can be basically used, or what kind of people that I want that expertise in this field can work with me to make this happen. So again, yeah, that's like a different line of research that I'll be working on to just going into one typical topic. Yeah.
Last question.
Yeah.
Why do you think that someone would prefer your this technology-based product over a OTC product? Let's say if I'm suffering from some insomnia, which is very easily curable by medications, why
Yeah.
should I go for a microcapsule-based product,
Yeah.
the one which you
Well,
are trying to develop?
yeah, sure. So basically, my program has a really small initiative. It's a very simple sentence of me not going over to the field of OTC. I would not make a microencapsulation technology clothes which basically deals with, for example, digestion, or maybe would help you with fever, because those are really small problems. I focus on life-term problems. For me, cancer is a big thing. For me, lifelong skin conditions is a big thing. Substance use disorders, addiction is a big thing. So, Medicator is basically offering localized and targeted delivery with therapeutic agents directly through the skin, which is again a thing to think about, that these are a long-term disease focused areas. I'm not focusing on something that can be cured in within few weeks or within few months. This is something that person goes through months and years, and to ease that procedure, to ease that method, medical helps it with the comfort. That's it.
Thank you. Thank you from my side.
Thank you so much.
Yeah, I have a couple of questions.
Yeah,
So
thank you.
how do you visualize it? Like you visualize this as a disposable garment or it's a reusable garment?
Okay.
When you talk about fabric.
Yeah. So basically, the only reason I made it Medicator, and why I came up with this thing called as fashion industry, because I needed to solve fast fashion as well. The one statement that I mentioned in my PPT as well is maximizing efforts, maximizing the efficacy, and minimizing the waste. Minimizing the waste is basically like the fast fashion. Well, let's think about H&M or Zara. These
No, no,
brands.
my question is very simple. My question is
Yeah,
very
yeah.
simple. You consider this as a disposable solution or a reusable solution as of now?
It's a reusable solution. Again, we are using recycled, we are using defected pieces from these brands. We buy these pieces from the brands and we recycle it. We come up with this technology into them. These drugs will end by 1 to 6 months of efficacy. And once that's happening, we will come up with reloadable technologies, which is again a different thing to work on. But for me, at this moment, it's reloadable and it's reusable from the recycled product.
Okay. So how do you visualize a cap for schizophrenia, a jacket for melanoma, a trouser
Well,
for
yeah.
rheumatism? Is it something what goes in your mind?
But visualizing would really depend on from where the drug would penetrate faster and from where the efficacy is more. Why have I focused on skincare disorders? Is skin has already lost its barrier. So for me, a tight jacket would be a great solution. So the penetration is more easier as the sweat comes in. Penetration becomes more easier for the drugs to go inside and get absorbed. So it depends on disease to disease how the clothes can.
I think the basic, yeah, basic principle you are probably overlooking is if you want to deliver the drug through skin, it has
Yeah,
to be in contact with the skin.
yeah, it has to be contact with the
Yeah,
skin.
yeah. So
That's
when you talk about a fashion statement,
yeah.
so you may have layered garments and they may not come in contact with the skin, right?
Well,
So there are
yeah,
two different aspects. So one is comforting garment is a different thing, and you are trying to deliver medicine through a garment which has to
yeah,
be coming in contact with the skin. This is a very complicated proposition or a
okay,
solution for therapy,
yeah,
drug-based therapy. So, because there are even we talk about drug delivery system, there are thousands of factors which
yeah,
decide, right, from the dose
yeah,
which can be delivered, the drug that can be delivered, and the
yeah,
effectiveness and safety of such delivery systems. So I
yeah.
think this is not going to be. Have you looked at the customers or the patient's persona when you are trying to design this delivery system? So who, like, is 10% of the affluent class will wear your fabric, or it is going to be helpful for the patient at large?
Well, at the initial stage, it really focuses on hospitals. As in my opinion, these are for the hospital clothes, for the hospital bed sheets. which are really in contact with the patient's skin. But as we go ahead with the research and as we come up with personalized program, I think it would be open for the lay person as well, for the people in the underprivileged and underserved area as well. But for the initial stage, it's for the major companies, which is the government sectors and hospitals.
Okay. Thank
Yeah.
you.
Thank you so much.
Thank you, judges, for your valuable questions. Now let's move on. Code number IIC 006. I request you to turn your camera on.
Hello everyone. We are IIC 6. Our topic for today, or our proposal as such, is green biomanufacturing of hypolipidemic cordycepin using engineered yeast and agricultural waste molasses. To give you a gist of our project, it is that we will use agricultural waste, which is molasses, as a sugar source in fermenting a genetically modified yeast, which is S. cerevisiae, to produce hypolipidemic cordycepin and yeast biomass. This yeast biomass is further recycled as a yeast nutrient medium in microbial purposes and as animal feed or food additive.
Okay, so to start off with the introduction, 67.8% of Indians are aged between 25 and 64 years old. And as we all know that with age, the risk of having high fat levels in the blood increases, and that leads to high risk of having atherosclerosis. More so, the Lipid Association of India has stated that Indians over 45 years of age are at a high risk of having atherosclerosis, and Indians have atherosclerosis a decade earlier than the Western population. So this means that over half of the Indian population is at risk of having atherosclerosis, and thus there is a need of having drugs, hypolipidemic drugs, which can decrease the high lipids of the blood. So the drugs that do that, the most common example are statins. So although statins are effective over the years, a lot of side effects have been reported, like loss of function of muscles, type 2 diabetes, then stroke, and hepatotoxicity. Thus, there is a need of other alternative or adjunctive drugs which can help in decreasing high lipid level while being safe. So one such compound which has this hypolipidemic activity is cordycepin. So there's a fungi called cordyceps which produces a lot of bioactive compounds which have various benefits, as you can see in the figure. So the one that we want to focus on is anti-hypolipidemia, which means that it can decrease high blood lipid levels. The one main bioactive compound that has this activity is cordycepin. Along with that, along with being hypolipidemic, it also has anti-inflammatory and anti-tumor activity.
So we did a brief market analysis of both cordyceps and anti-hypolipidemic drugs to get an idea of how much the market value is and the compound annual growth rate. So we could not include cordycepin because it is not as such produced in the market, and cordyceps is the focused product. So through this, what we got to know is cordyceps and anti-hypolipidemic drugs have a market size in millions and billions, and the compound annual growth rate is estimated to increase over 8 years at an exponential rate, and the average cost of cordyceps is greater than anti-hypolipidemic drugs, with a steep decline in statins. So some products of cordyceps that are available in the market are aiming towards potential health benefits and targeting different factors such as anti-aging and anti-inflammation.
Okay, so now that we've introduced the topic, the rationale of our topic is essentially to produce the compound cordycepin, which although is produced by cordyceps, it is produced in low amounts. So through the proposed green biomanufacturing method, we will use a genetically modified Saccharomyces yeast, which will use agricultural waste molasses to ferment it into cordycepin. The advantages of this method is obviously that it is highly sustainable. It also has high yield, and the fermentation cycle of growing yeast is also very less when compared to the cordyceps fungi. So to start with the methods, the first thing that we want to do is genetically modify our yeast, that is Saccharomyces cerevisiae. So Saccharomyces is a GRAS status, which means that it is safe for consumption by humans. So to start off with the modification, we first of all have to extract the genes which synthesize cordyceps from the fungi cordyceps. So this cordyceps can be grown in the lab, as you can see in the image. And once they form a fruiting body after around 60 days, these cells can be lysed and the genes which synthesize cordyceps can be extracted like CNS1 and EA1 and they can be introduced into S. cerevisiae. So now once we have our genetically modified yeast, we have to grow it. We have to understand which conditions are the best for its growth to get a high yield of cordyceps. So there is a method called one factor at a time, OFAT, which will be used to check the best temperature, aeration, the best molasses concentration, and the pH for the highest yield of cordyceps that we can produce. Once that is done on a small scale, it can be scaled up to a 5-liter bioreactor and so on.
So to after the fermentation process, to obtain the cordycepin, there are a series of processes that will take place. So after fermentation, the culture broth will contain cordycepin, which is our main product, yeast cells, and other metabolic byproducts. So the sub-processes are as follows. Centrifugation or filtration will take place to separate yeast biomass from fermentation broth. This is followed by solid phase extraction, where we will extract the cordycepin from the broth using resins or adsorption columns. The cordycepin is further refined using solvent, precipitation or HPLC, and cordycepin is then lyophilized or vacuum dried to be obtained as a powder. After this, to obtain the yeast biomass in the purest form, the leftover yeast biomass, which is the byproduct, is recycled. So after separating from the fermentation broth, the yeast biomass will undergo lysis, where the yeast cells are broken down and are converted to yeast extract. Following this, centrifugation or filtration will take place to remove insoluble debris and to obtain nutrient-rich yeast extract. After this, we will follow it with evaporation or drying to produce yeast extract, which is a nutrient yeast medium for microbial purposes and food additive or animal feed ingredient.
Okay, so now that you've understood our proposal, although it seems easy on paper, there are a lot of hurdles to overcome. So the first hurdle is to manage the variability of the agricultural waste molasses. Because there are a lot of sources and variation in the amounts of sugar in the molasses, so we have to standardize that. Apart from that, it can also have some adulteration. So molasses has to be pre-treated to take them out. Then the yeast that will be used, it is a genetically modified organism. So that has to be stabilized as it grows. And moreover, as the yeast grows, it will produce some toxic metabolites which have to be taken out so that the yeast can grow properly. Once all of that is achieved and other unexpected hurdles, in the future, this method can be scaled up to industry. And once we reach industry, the cordycepin can be formulated into a hypolipidemic formulation like a drink or a tea, etc. And then it can go for animal and human trials, along with, in comparison with other drugs like statins, and then eventually it can be used to produce other products like cosmetics.
So in conclusion, cordycepin, which is a main compound of interest, bioactive compound, it is a hypolipidemic compound and it is produced using S. cerevisiae yeast, which has been genetically modified for efficient cordycepin synthesis. We use molasses as a sugar source of fermentation substrate. It is a low-cost byproduct from agriculture waste, and yeast biomass is then recycled into yeast extract, reducing the waste, and this whole process ensures that it is a sustainable one and it combines economic feasibility with environmental benefits for scalable production.
Thank you.
Thank you.
I now request the judges to put forth the questions.
Yeah, from your presentation, I understand that the cordycepin products are already marketed, right? Some of them are already marketed, correct?
Just for cordyceps.
So what is that source or what is your comparator basically? As in when you are saying that this is a green biomanufacturing for cordycepin, what are the other processes that are already available and to whom you would be actually comparing this particular process?
So, sir, recently we had a guest lecture where we learned that sandalwood takes normally 20 years maximum to grow and to get the sandalwood oil. But the guest lecturer said, he's from CMAP in Lucknow. He said that the oil can be extracted within 6 days using yeast, like it is genetically modified. So, in a similar manner, we want to modify our yeast of interest to get cordycepin. So that
So
is the
currently,
comparator.
you mean to say currently cordycepin products are not available?
Cordycepin as such is not available in India.
Mhm.
Synthetic cordycepin is available. We are saying green biomanufactured cordycepin is not available.
In one of your slides, there was a mention of standardization actually, because I was pretty curious to know how are you gonna standardize it basically when you know that there are so many variations in terms of the molasses.
So about that, there are methods which help to standardize the particular concentrations that we have to use. So it applies to all factors, whether it be temperature, concentration of molasses, etc. So the method that we'll use is one factor at a time, and we will source our molasses from one particular industry. So that will help us to standardize the our source and the concentration.
Yeah, that's it from mine. Thank you.
Okay. So how will you take care of the demand supply if you are going for a green manufacturing, uh, where you have shown a picture of the insect, right? So how uh, have you studied about the demand supply?
So,
So, since the idea, I'll go ahead.
yes, please.
So, ma'am, the idea that we are proposing as such is specifically for a hypolipidemic drug. So, for that, there is already a market, like we have statins already, right? So, for that, we already have a market. But since there have been a lot of side effects for it, the doctors and patients want a new drug which has fewer side effects. So, there is a demand for this. And as for the supply, um, since the idea that we propose is based on a microbial fermentation, the supply would be quite easy because growing cordyceps takes around 2 to 3 months. Whereas for yeast, producing cordycepin through the yeast biomanufacturing takes only 2 to 4 days. So that way, our supply would also be able to keep up with any increasing demand.
But your fermentation is dependent on an organism, right? It is dependent on a living cell, correct? So how is the population of that particular insect? Will it be able to give you the large production of your product?
Yes, ma'am.
Is my
So
question clear to you?
actually, yes, ma'am. Yes. So studies have shown that yeast can produce this particular compound, like the yields that have been obtained, they range from 2000 mg per liter to 3000 mg per liter as well per day. So the amount that will be produced is quite high, even with just a small amount of organism. And obviously, that's just one bioreactor. At an industry scale, we'll have like maybe 2 or 3 bioreactors. So the yield would be definitely higher. Is the explanation okay?
Yes, thank you. Okay, that's all from my side.
Yeah, so I wanted to understand some of the aspects in this. Is the process of cordycepin manufacturing established by fermentation?
Yes, sir.
Okay,
So there is a
good.
similar study
It is,
on
right?
Yerovia
So
lipolytica,
it's the
which is
only,
another
it
yeast.
is one,
Sorry, sir.
sorry, go ahead.
Okay. So there's another study on Yerovia lipolytica, which is a ascomycetic yeast, And there they established this fermentation method using a bioreactor, and the references which we are giving are according to that. So there, it is an established method.
Now, are you familiar with the regulatory aspects of genetically modifying the yeast that you are going to do?
Yes, sir. So there will
Yes.
be like we will require some regulatory confirmations before we try this, and like GAC, IEC, and all. So, but that is genetic engineering is a one-time thing. So if we get that approval, we can proceed with the work.
Okay, and
So to add on, sorry. So to add on, since the yeast that we'll be modifying is not for consumption, so the
Yes, sir. You may continue.
Thank you. Thank you, judges, for your valuable questions. I now request code number 007 to be ready and turn on your camera. We'll be presenting your video soon.
Hello everyone. So first of all, I would like to thank the organizing committee for providing me this opportunity to present my concept in Innovative Idea Challenge category. My identifier code is IIC07. So today I will be presenting my topic, that is development of an anti-tobacco smokeless products. So as we know that tobacco use is a major risk factor for many chronic diseases, including cancer, lung disease, cardiovascular, strokes, etc., leading to nearly 1.35 millions of deaths occurring every year in India. When it comes to tobacco consumption forms, so basically they are available in two forms. The first one is a smoking tobacco products, and the other one is referred to as a smokeless tobacco products. So these are the depiction of the smokeless tobacco products as well as the smoking tobacco products. When it comes to statistics, so basically as per the Gates, that is Global Adult Tobacco Survey, nearly 29% of the adult, which correspond to 267 million adults, they are users of tobacco products. So as the following figure depicts, the following pie diagram depicts the bifurcation of those 29% of the adult, indicating that nearly 7% they are smokers, whereas 18%, which falls under the major category of those 29%, they are consuming the smokeless tobacco products. So basically for those 7% population which are addicted to these smokeless products, various smoking cessation products are available in the market, like Nicoderm, Nicorette, Nicotex gums, patches, etc. Whereas for those 18% population which is addicted to the smokeless tobacco products, no specified products are available for those particular category, as well as the drawback of the existing product has led to the development of this particular invention. So when it comes to problem statement, tobacco use is a complex interplay of habit, sensory satisfaction, and chemical dependency. The drawback relate with the existing cessation products like gums, lozenges, or patches are they addresses the nicotine dependency but fails to cater to the sensory and habitual aspects of the smokeless tobacco products. That's why there is a need of such products which not only assist in cessation but also mimic the smokeless tobacco products, providing psychological satisfaction and diminishing physiological dependency on tobacco. When it comes to invention, so basically the particular figure indicates the smokeless tobacco products like pan masala, gudka, which is available in the market, whereas on the other hand depicts the non-tobacco, which is a scientifically developed product that mimics the smokeless tobacco products in terms of its taste, texture, and feel in order to enhance the compliance rate of that particular addicted person. When it comes to the working, so basically this non-tobacco, which is a scientific product, it is based on the principle of NRD, that is nicotine replacement therapy, which is based on the controlled administration of nicotine, as well as it aims to both to stimulate nicotine receptors, thus removing craving and withdrawal symptoms, and to reduce the number of nicotinic receptors. When it comes to the objective of the invention, so basically the primary objective of the present invention is to design a product that mimics or that is very much similar to the smokeless tobacco products like gutka, khaini, zarda, pan masala in terms of its taste, in terms of its feel, texture, and way of consumption. So basically, when the person consumes this particular product, so its compliance rate is enhanced in compared to the other products. The second objective is to develop a product that is safe, cost effective, and industry feasible. When it comes to materials and methodology, so basically we have used the nicotine polychloride, which is the source of nicotine, as well as the API in this particular product, whereas the other excipients are chosen from the following classes, that is diluent, binder, flavoring agents, alkanizer, and sweeteners. When it comes to methodology, so we have opted its simplest methodology to develop a particular product. That is, the step number one is we will be taking the PVP, that is PVP K30, which is acting as a binder. We'll be dissolving, we'll be preparing the binder solution. The second step is we will be sieving, or we will be shifting the nicotine polychloride, MCC, lactose monohydrate, mannitol, sucrose, and sweeteners through a mesh size of 30. Further, we will be adding the binder solution in this particular mixture to obtain a wet mass. While adding the binder, we have taken the care of that it should not be get wetted excessively. Then we have dried, then we have passed that particular wet mass through a mesh size of 6 or 8, and we have kept them granules for the drying process. After drying, we have shifted the granules to a mesh size of 30 screen, and then we have added magnesium stearate and flavorant, which is acting as a lubricant, and then we have mixed thoroughly. Then we have taken these granules for the compression process. Further, we have taken those particular tablets and passed through a milling machine to obtain the particular texture of our product. When it comes to feasibility and scope, so since the formulation incorporates the FDA approved excipients which are safer, and the manufacturing process which is used is a straightforward, leveraging existing equipment and infrastructure in the pharmaceutical industry, making it a industry feasible process. Whereas when it comes to scope, so basically since we have already seen that 29% of adults, they are consuming tobacco, so even a 5% market penetration translates to millions of users switching to a safer alternative. So these are the silent features of a non-tobacco, that is scientifically developed product which looks like smokeless tobacco products like gutka, pan masala. The dose of the products is available as per the consumer habit pattern. When it comes to benefits, socio-economic well-being of the society, for all age group of addicted society, improved compliance due to the equivalent format as that of habit forming product. So hence we have particularly focused on this particular parameter, that we will be improving the patient compliance by developing a product that mimics the smokeless tobacco products in order to overcome its psychological dependency. When it comes to competitive landscape, since non-tobacco is a unique product in the domain of the smokeless tobacco products, cessation products, so there is no direct potential competitors. But when it comes to some of the products which are available in the market, that is nicotine gums or patches, lozenges, etc. So when it comes to parameters, that is cost. So basically the proposed product will be lower cost in comparison to the products which are available in the market. The second one is the compliance rate. So basically we are mainly focusing on this compliance rate since it is mimicking the smokeless tobacco products. So when the person consumes our product, so it will feel the same texture, same feel, and same very much flavor when he or she, the addicted person, will be consuming this particular product. When it comes to idea, so basically the proposed concept has been granted patent for the idea recently in December 2023. When it comes to discussion and conclusion, so basically the non-tobacco represents an innovative product with a smokeless tobacco product sector which is specifically designed to curtail and eliminate nicotine dependency. The main objective of our project is to design a product that mimics smokeless tobacco products, providing psychological satisfaction and physiological dependency on tobacco. So at last, I would like to conclude that non-tobacco is not just a product, but it's a movement, a commitment to saving life, reducing the burden on the tobacco related disease and promoting public health. So these are the references, and thank you, sir.
I now request the judges to please present with your questions.
In your one of the last slides, you mentioned about the compliance, and I couldn't actually understand it. What compliance it is actually? You said the better compliance over the other products, right? Yeah.
Code IIT07, you are not audible. I'm sorry, but code IIC07, you're not audible.
Participants, please hold on. There is some kind of glitch. We will soon continue with the questions in about 5 minutes.
I request the judges, if it is okay, we can do the question answer session later on and move on to the next video.
Sure, sure. I think that works.
Yeah. So code number is IIC008. I request to please turn on your camera and we'll be presenting the video soon.
Greetings to everyone. So here my project aims to develop a method for real-life application of an AI active fluorophore for sensing of trypsin in urine samples. Why trypsin? Trypsin, as we know, is a pancreatic enzyme where it is responsible for digestion of food to break down larger protein molecules into smaller peptides. The average concentration of trypsin seen in human urine is 0.115 to 0.350 micrograms per ml. What if the trypsin is generated in excess? So in a normal condition, excess trypsin is regulated by pancreatic secreted trypsin inhibitor, which is a naturally occurring substance. Therefore, it acts as a first line of defense to protect the pancreatic acid in our cells. However, if trypsin activation exceeds the capacity of PSTI, it leads to a series of events where finally it leads to autodigestion of pancreas. Therefore, it leads to initiation of acute pancreatitis. However, deficiency of trypsin is seen in chronic pancreatitis, where the thick mucus layer obstructs the pancreatic ducts, causing inflammation of pancreas over time. In this condition, trypsin activation occurs too early, which attacks the tissues of pancreas, further leading to scarring and shrinkage of the pancreas, resulting in insufficient generation of trypsin. Seeing this, there are current detection strategies to monitor trypsin levels, which includes ELISA tests, electrochemistry-based methods, colorimetry-based methods, etc. These methods show certain limitations like complex design, high cost, limited sensitivity, and so on. Also, the reported fluorescence-based methods struggle to operate in the competitive biological matrices, therefore impeding its prospect in the real application. Therefore, we saw an evident demand to develop a trypsin sensor which can operate efficiently in biologically competitive media. Here, my design strategy includes histone as an enzyme-specific substrate. Reason being, it contains high arginine and lysine content, which is susceptible to cleavage by trypsin. This sensor scheme operates on the principle of aggregation-induced emission, which is a phenomenon utilizing a certain class of fluorophore which emit light only in their aggregated state. As seen in the right-hand side structure, the intramolecular rotations of the dye molecule are free to rotate in the aqueous medium, therefore giving non-radiative processes. In the aggregated state, this intramolecular rotations get blocked and giving the emission enhancement. This is my scheme of sensor where, as I said, histone is my enzyme-specific substrate and BSPO-TPE is the free fluorophore. Due to electrostatic interaction, this BOTPE forms an aggregate on the surface of histone, forming a supramolecular self-assembling system. This is the emissive state of the complex where we can see a turn on in the system. In the presence of trypsin, it acts on the site of cleavage of histone molecule and releases the free fluorophore, going back to its non-emissive form. Here we can see in the steady state measurements as we gradually increase the concentration of histone, there is emission enhancement, which is so strong that we could see a 364-fold emission enhancement. This was supported by the absorption studies where there was a rising offset at the longer wavelengths. This is due to the scattering effects of the aggregate that is formed. We can also see a turbidity in the solution due to this aggregate formation. The lifetime studies here show that there is a gradual increase in the trend where blue line is showing the maximum aggregated state of the complex. This sensor was evaluated for various environmental conditions to check its stability, starting with ionic strength effect, where we can see NaCl is able to screen the interaction of BSPO-TPE and histone. Here, on gradually increasing the concentration of NaCl, there was disassembling of the BSPO-TPE aggregates from the histone surface. Also, in the absorption measurement, there was a decrease in the offset, thereby confirming that there was an electrostatic interaction between BSPO-TPE and histone molecule. Coming to the temperature studies, here we saw that on gradually increasing the temperature, thereby providing more energy to the system, there was disassembling of the aggregates from the histone surface. The lifetime studies also shows that on increasing the temperature, there were faster decay traces. pH studies shows that the sensor is quite operative between a pH range of 4 to pH 8, which covers the biological pH of 7.4, thereby showing that it can be efficiently utilized in the human urine. Selectivity studies shows that amongst various enzymes present in the body, it is highly responsive towards trypsin. Coming to the later half of the scheme where trypsin is able to cleave the histone substrate. So here we could see that on gradually increasing the trypsin concentration in the system, rate of hydrolysis of histone molecule was increasing. Lifetime studies here were done by using a specific trypsin concentration and studying it over a time scale. So red line is showing the aggregated state, whereas the blue line is showing the cleavage done by the trypsin to the histone molecule. Um, Getting motivated by the earlier studies, we saw we saw this effect in 20% urine matrix, where we could see the similar trend by increasing the trypsin concentration. The rate of hydrolysis of histone molecule was increasing, and we could see a spanning range of 0 to 0.42 micromolars. Now, this sensor scheme can be applied for bedside detection of trypsin, where the patient urine sample can be treated by the sensor probe, that is PSP, TPE, and the histone. And further, it can be used in the point of care device, where it can give a quantitative trypsin value. Therefore, we conclude that we could achieve an LOD of 0.91 nanomolar, and this sensor scheme is quite simple, cost-effective, and sensitive approach compared to the traditional methods. These are my references used for the project. Thank you.
I now request the judges to start with the question and answer session.
So you in the last sentence you mentioned about the traditional methods. What are these traditional methods actually?
So traditional am I audible first of all?
Yes, you are audible.
Okay, sir. So traditional methods include ELISA-based methods or colorimetry-based methods, electrochemistry-based methods, and so on.
And what are the, like you mentioned, the trypsin concentration connected with the acute pancreatitis, I believe, right? The pancreatic condition. Are there any other applications for this?
Applications as in, sir?
As in the evaluation of trypsin actually, or the trypsin concentration, can it be also used in any other diseases or any other conditions?
Trypsin actually we can see monitor during pancreatic cancer or acute pancreatitis. Also, it is seen in chronic pancreatitis or cystic fibrosis and biliary cirrhosis. So, in these conditions, trypsin levels are usually monitored.
Okay, that's it from my end. Yeah, thank you.
Thank you, sir.
Srinivas sir, would you like to ask or should I ask?
Yeah, go ahead. I'll ask later.
Okay. Okay. Have you standardized this method that you are using against any reference?
This method project is actually not completely done because we need to do the validations compared to the traditional methods that are available, as well as we need to validate in the clinical samples as well. So these are currently done in laboratory based in buffer systems, and also in the human matrix that was treated.
And have you checked the effects of any other impurities, dust particles, which can affect the sensitivity of your sensor?
No dust particles or any impurities were seen. Only we checked the stability of the sensor in various conditions, like I mentioned in the presentation, like ionic effect strength, temperature conditions, which could normally affect the sensor in the real conditions. Impurities as such, human urine won't involve. Other things are like other enzymes in the body that could affect the sensor system. So selectivity studies were done in that respect, where we could see that only trypsin was it was efficiently giving the response.
And have you done any confirmatory test for the complex that is formed?
Confirmatory tests were done using steady state measurements, absorption studies, as well as lifetime measurements, TCS-PC measurements, basically.
Thank you.
So, can you tell me brief about the construction of your sensor?
So So the sensor scheme involves the BSPUTP as a fluorophore. So in its aqueous state, the intramolecular bonds of the molecule are free to rotate. Therefore, they do not give any radiations. But once it has a co-component which is oppositely charged, it can form an aggregate on the surface of the substrate, that is histone as I mentioned. So this aggregate now will give an emission enhancement, and it can be seen that there is a turn on in the system. In the presence of trypsin, if seen in the patient urine, this trypsin will act on the cleavage site of histone, that is carboxyl.
My question is not. My question is not on the mechanism. My question is more on the physical sensor.
Could not connect, sir. Sorry.
So, are you building a physical sensor which can be dipped into the urine sample, or you are just making these reagents which like?
No, sir. So these are reagents are commercially available. So I would be developing a sensor where the patient urine can be treated with the fluorophore followed by the substrate, and it can be monitored by giving a quantitative value of trypsin directly in the point of care device that we would be constructing actually.
So what is that device? Like, how would it appear? Will it be in the form of a strip? Will it be in the form of
the device will be based
holder?
on. Yeah, so as I mentioned in the slide, the device would be having LED laser sources. It will also have a PMT detector in it and a sample holder where we can place the cuvette filled with this solution.
Okay, so cuvette is where you are going to add the fluorophore and the sample. Is that so?
Yeah, so patient urine, the fluorophore and the substrate. So this all will be in the cuvette and it would be placed in the sample holder. So it would be a miniature setup with the assembling of these devices like LED laser sources, PMT as a detector.
Yeah, but it's more like a miniature fluorescence spectrophotometer, right?
Yeah.
So do you call it as a sensor?
Yeah, it would be a sensor because it is based on the phenomenon of aggregation induced emission. This is a concept where we can actually sense the trypsin values. It will give a quantified value where we can, sorry.
How will it be a point of care device?
It would be used in hospitals for bedside detection of trypsin, like the patient urine can be collected and it can give the trypsin value.
All right, so this is not a point of care, right? It's not for personal use.
Yeah, it wouldn't be for a personal use. Sorry.
Right. Okay, thank you.
Thank you, judges, for valuable questions. I now request IIC 07 to please be on camera and request the judges to please start the question and answer session.
Hello. Yeah, hi. Yeah. So my question, the first question that I had asked was about the compliance thing that you mentioned as such in the last slide. What is that compliance with respect?
Sir, first of all, I'd like to apologize for the technical issue. So basically the products which are available in the market for the tobacco cessation. So basically, for example, if a person is consuming smokeless tobacco products like pan masala or gutka, so he's normally taking parking between the cheeks and the teeth. When it comes to gums, so basically its mode of administration is you need to bite and then park. So basically, when it comes to tobacco consumption compliance rate, so we have developed the product which will mimic the smokeless tobacco products. So when the consumer take up a product, he need to do the same to do for the smokeless tobacco products. So because it mimics the smokeless tobacco products. So when he or she consumes the product, so it will get the same feeling and sensation like that he's consuming the smokeless tobacco products. So in that rate, we have enhanced the compliance.
Okay, so it is more of the habit actually, or like the way you have
because
compared.
Yes, sir, because it has been already reported in certain literature as well as in certain reports that the tobacco is main of physical, psychological dependency and physical dependency. So the product they are psychological, hence psychological dependency, which is the main we can say the person which is responsible for the withdrawal symptoms. So we have overcome that problem through our product.
Right, and would that be the USP for the product as well?
Yes, yes, that's a usually product. So that is why, sir, the innovation which we have invented, we have also protected our invention. That is the USP of our product.
Okay, thank you. That's it from my end.
Okay. Shivanshu, sorry, I07, right?
Yes.
Okay. So have you done, you said that your product is exactly similar or you plan to make it similar to a marketed product like Gutka. So have you done any evaluation on the taste of the product?
And basically the product, when it comes to the taste and texture, so we have normally compare the texture of the smokeless tobacco product with our product. So basically, in terms of size of that particular texture, we have with the smokeless tobacco product. So it shows that the size is relatively similar to smokeless tobacco product. When it comes to taste and texture, since we have used the flavors which will mimicking the smokeless tobacco products, so in that sensation,
So what are the parameters on which you have you can claim that this product will be accepted by a user who is having tobacco regularly? How what
the first
basis
one
you say
is the it's
it is mimicking? What are the parameters for you to mention it is mimicking the said product? Can you give me the
the first one
pointers?
is the first one is the texture of the product, which is the product. The taste. The next point is the particle size
Correct.
distribution.
So
Since we
so
have
when you say taste,
evaluated the particle size,
when you say taste, so how have you evaluated the taste? Texture is visual, but how about the taste? Have you tried it on volunteers? Have you done some
given
volunteer
normal.
study?
Ma'am, actually, uh, we have tried on 23 people about the taste of the particular products. So it,
Okay.
it has the
So
taste which is similar to the smokeless tobacco products.
so when you say you have done the study on 23 people, uh, was it done as per the ethical permission? Did you have to take any permission or how was it? What was those 23 people? On what basis were they selected?
Actually, it was just a trial and error. It was just a trial to see that the product which we have made, whether it is
Mhm.
actually mimicking the product from the consumer view. So we have done it in a small location for product in comes in terms of its taste and texture.
Okay, thank you. Yes, sir.
Uh, so what is the need of this product?
So basically, the addicted person who is consuming the smokeless tobacco products, for those people, we have developed this product to reduce the tobacco addiction.
But there are better offerings in terms of lozenges and chewing gums and all.
Sir, will you please repeat your question?
I said there are better offerings already in the market, right?
Hmm.
So what is the need of making a nicotine Polacrylex tablet?
So basically, the products which are available in the market, they have shown the clinical efficacy, but when it comes to the habitual action of that particular consumer, so in that particular kind of case, well, means when the consumer is going on the therapy, so in the almost 80 to 85% for consumer, they get the withdrawal symptoms, they relapse and get again addicted to tobacco products. But when our product they will be consuming, so since it is mimicking their psychological, it is overcoming their psychological satisfaction that we are consuming the same product as our habitual, which we are earlier consuming. So in that compliance, in that context, it will enhance the rate and a compliance rate of the people.
Yeah, but what about efficacy? This has to be proven, right?
Yes, yes, sir. Basically, in order to see where we are under that clear preclinical stage, we have still not studied the preclinical stage of this particular.
No, I don't. Are there any preclinical models to study the habitual factors, that is chewing and spitting and the feel in the mouth? Do we have any preclinical models?
Actually, I have not particular. I will see that particular topic in after that.
Yeah. So basically, how you prove the features of your product is important. This is just a perceived idea, or you have started working. You are saying you have tested in 23 people also already.
We have just taken the feedback. Actually, we have not tested actually on that this is particular product which we are claiming. We have got the feedback from those people that whether this product is mimicking your product or not. So we have not purposely we have done that, but we have just to get a feedback from those volunteers. We have done that.
So which will be the products you will be comparing with no tobacco product? I mean, smokeless products. Which brands of gutkas or pan masalas, or what is your target?
So basically, we will be comparing our product with the products which are available. For example, Nicotex lozenges, Nicotex lozenges are available. Will be and it will be available in combination.
Those are not head-on comparators, right? They are related to nicotine products. But you are trying to have a habitual difference. You should have a comparator which has, for example, they are chewing gutka, and instead of gutka, they will take your product, right?
Yes, sir. Basically it's a medication which is aiming to reduce the tobacco consumption. So hence for this purpose, because in the literatures they have already reported that the people that currently products are available for the cessation, but the reported literature states that the consumer, the patient, they are relapsing because of that habitual action. So that's why we have aimed to overcome that habitual action. Once we have overcome the habitual, since it has already proven its efficacy, so it will be effective in terms of its treatment.
So just one last question. So what would be the duration of treatment when you want to do head-on comparison?
So, sir, basically the duration will depend on the consumer addicted profile. For example, if the particular person is consuming in a day 10 sachets of that particular product, smokeless products. So for those people, we will be going ahead with the higher dose of that particular product. For example, let's say it's a.
No, I think your endpoint is to kind of improve the compliance, right?
Yes, sir, because that is the.
So you have to have
yes, sir, that is
the studies for certain duration, right?
Yes, so that is the main barrier which we are trying to overcome, because once we have overcome that psychological barrier, so we will be moving ahead with the effective therapy for tobacco addiction.
Okay, fine. Thanks.
Thank you, sir.
Thank you. Now I would request code IIC09 to be present. Switch on your camera and let's carry out the last video for the day.
Greetings to one and all. My identifier number is 09 and title of my project presentation is Real-Time Fluorescence Sensor for Alkaline Phosphatase Detection via Reversible Pyrene Polymer Aggregation. Alkaline phosphatase is an enzyme which is present in human body and mainly found in liver, bone, intestine and placenta. The normal range of alkaline phosphatase in human serum is 40 to 160 unit per liter. But in certain disease like liver disease, bone disorder, kidney disease, there is change in concentration of alkaline phosphatase. This abnormal change in concentration serve as a biomarker for this disease. Hence, there is a need of detection of alkaline phosphatase. There are several methods are available, like traditional methods, ELISA, colorimetric assay, which have certain limitations like low sensitivity, environmental dependency, and high cost. To overcome these limitations, there are fluorescence-based sensing methods are emerged with promising alternatives to traditional method and provide high sensitivity, rapid response, and adaptability. But there are certain challenges with single emission sensors, like when there is a change in environmental factors like change in pH, salt concentration, which leads to change in response. Hence, to overcome limitations of traditional method and challenges from single emission sensor, we are adopting ratiometric fluorescence based sensor, which gives self-calibrated output, minimize error from environmental factors, and enhance the accuracy as well as reliability. In ratiometric fluorescence sensor, we are using supramolecular assembly, which is simple, sensitive, cost-effective, as well as easy to design. In supramolecular assembly, we are using a dimolecule and polymer. So here, dimolecule is a pyrene tetrasulfonate sodium salt, which is tetra negatively in charge, and polymer, which is polycation. Other than traditional and fluorescence method, there are commercial kits are available, which is based on colorimetric as well as on fluorescence base, which have higher cost as well as higher incubation time as well as assay time. In this, we can see 15-minute incubation time is required and it is microplate reader base. But this is another kit which required assay time 1 hour 10 minutes approximately. So here we can see there is a need of sensor which will give quick response with low cost as well as high sensitivity. So we are proposing sensor for paper-based LP detection in serum sample. Here we will use paper. At the center of the paper, we will embed probe molecules. That probe molecules will be PTS, PA, ATP. This on the probe molecule, we will load serum sample. In the serum sample, ALP is present. That ALP will hydrolyze some substrate from the probe molecule, and that gives change in color. This change in color we will compare with the color strip, and we can find out the approximately concentration of ALP from the serum sample. The benefit of this sensor system is it is operational in serum sample. It is easy to use by any biomedical workers. It gives quick response time which is less than 5 minutes with low limit of detection, and there is no maintenance as well as calibration is required. So in this system, we will use dimolecules. It will interact with the positively charged polymer, that is PA, and it will form complex. So formation of PTS PA aggregation is the first step of the sensor. In this system, it is
aggregated because of the electrostatic interaction which is occur between the negatively charged dimolecule and positively charged polymer. When in this aggregated stage, when we will introduce ATP, which is trinegatively charged, and basically it is substrate for enzyme alkaline phosphatase. So after introduction of ATP in the aggregated stage, it will disrupt this aggregated stage of PTSPA and it will change in monomer aggregate equilibrium. Here we can see the black dash line is of monomeric dimolecule, that is PTS. It gives fluorescence at 385 nanometer. Whereas when this dimolecule interact with the polymer, it gives fluorescence at 500 nanometer. This separate band is arise because of the pyrene molecule which is present in the dye. So in the literature, it is known that pyrene is known for this new band, which is known as exime. So this band is only occur in when pyrene is in aggregated stage. So when in this aggregated stage we will add ATP, this band will disappear, and because of the disruption of PTS-PA complex, there is a new band will appear, which is at 385 nanometer. So the new band, which is almost similar to the monomeric free dye molecule. This suggests that ATP is competitively binding with the PA. To support the fluorescence study, we perform lifetime. In lifetime, we can see this blue decay is of free dye, which is having decay lifetime value 10.5 nanoseconds, whereas the aggregated stage of PTSPA having decay lifetime value is 36.3 nanoseconds. In aggregated stage, when we will introduce ATP bring this aggregated stage decay to the similar to the monomeric stage of the dye molecules. Hence, it indicating that the ATP is breaking the PTS aggregate form of PA. So now, as I said, the PTS, PA and ATP will be our probe molecule. Here, when we will load the serum sample, the ALP which is present in serum sample that will cleave ATP, because ATP is a substrate of ALP. So this complex gives us higher specificity, because enzymes it is similar to the enzyme substrate complex. Now, after loading of serum sample, the alkaline phosphatase will cleave ATP in smaller fragment, and because of this reassociation of PTS-PA complex we will see. So in this we can see that this blue color band, which we already saw, here, this blue color band we will see now in this stage. Here we can see that after addition of ALP, this PTS-PA ATP system cause gradual recovery of the PTS-PA aggregated band at 500 nanometer. Means this band will again disappear after addition of ALP, and the band this at 500 nanometer it will again start reappearing because of the PTS-PA aggregation. This complete system we perform in the buffer, and with the LOD is 63.3 micro units per ml. So this suggests that ALP is hydrolyzed ATP and pave the way for reformation of the PTS-PA aggregate. To support the fluorescence study, we performed lifetime here, and we saw that this amplitude of slower component that is increased with the increase in the ALP concentration of the PTS-PA ATP system. And it is suggesting that the cleavage of ATP by ALP reform the PTS-PA complex. Hence, this system is offering a real-time monitoring of ALP activity. Here also, we can see that this is a free dye molecule. When it is in aggregated stage, there is a change in color. Also, in this condition, when we will add ATP, there is a change in color which is similar to the free dimolecule. And the fourth image is of when we will add ALP. So this color and aggregated stage of PTSPA is almost similar. Now, to perform a selectivity test, we use different enzymes as well as component from the serum sample, and we perform selectivity tests with similar parameters, similar experimental parameters to all enzyme as well as component, and we saw higher activity for alkaline phosphatase. So here we can see this is a higher selectivity for alkaline phosphatase compared to the other enzyme as well as component. Now this system we will perform in human serum matrix, and we saw that there is a good response in this matrix with LOD 936 microunits per ml. In conclusion, the paper-based sensors are inherently scalable, portable, quantitative detection with high precision, high selectivity in complex matrix, the reversible switching between the monomeric and aggregation emission state of PTS-PA system upon ATP addition and subsequently ALP hydrolysis serves the core sensing mechanism for ALP detection. This system shows good response in human serum matrix as well as high selectivity, sensitivity, rapid response, and commercial available probe molecules makes this system unique. Thank you so much. Thank you to SRC organizers and BNCP College. And the thank you to my all team members. Thank you so much.
I request the judges to please start with the question and answer session now.
Yeah. So what is the cost that you are looking for this kind of system? You mentioned about the time required actually to conduct the test. Those were your two comparators. One was the time required and the other was cost. But you showed us the time required for conducting the test. But then what about the costing part? Because that was a major, I would say, differentiator.
Yes, sir. So for cost, we are all probes are commercially available, and we are using paper. So we are for this paper, we are using different types of paper and including Watson filter paper. So till now we are proposing this sensor system. So we are not calculated cost yet, but we are thinking it is not more than the cost which is we are giving for the commercial kits as well as for traditional methods.
But then can you approximately let us know what may be the cost actually?
Sir, if I'm calculating, if I will calculate the approximately cost, then I hope it will be not. If we are proposing this in large scale, then it will be not more than 10 rupees. But means I'm talking about when we are in large scale production.
Okay, that's it from my end. Thank you.
Thank you, sir. Sorry, ma'am, I'm not able to hear you. Ma'am?
About this concept, what was the literature that you did and you realized that you have to work on this idea? What has created that idea?
Ma'am, pardon me, but I'm not able to hear your initial part of the question.
Okay, what was the problem with the existing ones that has made you think of this idea? So what was your problem statement?
Yes, ma'am. Ma'am, as lifestyle is changing, there are diseases are arising. Like people are changing with the foods and other lifestyle. In this, there are lots of diseases are arising in the liver, kidney disorders. And in that, specifically, the alkaline phosphatase is considered as a biomarker. So there is a need to detect this alkaline phosphatase in early stages as well as in quick response. So, we are proposing this detection method that will be. Ma'am, am I giving means? Am
Yes,
I
yes, I'm listening to you. Yes,
satisfying?
yes, yes, I'm listening to you. So,
So
what will be
for
the
the
paper used?
sorry,
It's a
ma'am.
paper based, right? So, what will be the paper used?
Yes.
You have mentioned paper based.
Yes, ma'am. We are now we are proposing, and in this initial phase, we are trying the cellulose-based paper, and there are we are trying the actually we are open to different types of paper, and then we will find out that which paper subject will be best to perform with this system. So we are trying initial phase. We are trying the Wortman filter paper, and other filter papers are available.
Okay, so you have estimated that it could be not more than 10 rupees. So, how have you estimated this price?
Ma'am, if we are going to produce in large scale, then there is we require only probe molecules, and these probe molecules are commercially available. So that commercial available probe molecule as well as the cost of the paper, only this much cost we will require. Hence, I'm thinking it will be not more than the 10 rupees.
Thank you.
So ALP is the enzyme
Yes, sir.
which is costly. In the ALT is enzyme which is costly. If we see in the some of the sensor system, and that ALP we are detecting, and it is naturally we are getting through the sample. So the cost will be reduced by itself.
Thank you. Yes, sir.
Thank you, ma'am.
Yeah, so I see that you did some concentration dependent response study, but that is in a liquid phase, right? Using
Yes,
fluorescence spectrophotometer probably. Was it a fluorescence? You use fluorimeter for that?
yes, sir. Yes,
Okay. And now you are coming up with a paper-based uh,
yes,
system. So what would be the detection
sir.
mechanism?
Yes. So on the paper we will load this probe molecules, and we will allow to dry it. So after drying we will add when this sensor system will be ready, we will allow with real matrix sample, and we will once the load the color change we will observe. But in this I use a dye molecule, that is PTS. The dye molecule is excited at UV range, but they give emission in aggregated stage at 500 nanometer, which is in the visible range. So that change in color we can easily observe by naked eye. And this system, it's in the market. It's already perform for detection of quality of raw milk. In my video, I added this photo of the paper-based filter paper-based sensor for quality of raw milk.
No, no. My question is, in your case, so it's just visible detection, right? So by naked eyes, you will
Yes, sir.
see the color developed, and you will decide the range of
Yes, sir. Sorry.
range of the.
There is
And
one
you
method that
mentioned it will be.
yes, we can see. Sorry, sir.
You showed a comparison with the pH paper, right? Color differences. So you can have a
No, sir.
semi-quantitative
I'm not.
indication of.
Okay. Am I sorry, sir? Can you please repeat your question? I think there is.
You showed that it will be a color comparison with the standard color codes, right?
Yes, sir.
So it's not going to be a quantitative test. It will be a semi-quantitative test.
Uh, Sir, here we are in the initial phase. We are proposing this that either we can go with the semi-quantitative test, like comparison with the color strip, or there is one more option that we can use. We can integrate this system with the smartphone, so that on the basis of the smartphone digital colorimeter, we can detect this change in color and how much color is. On that basis of we can find out the quantity, but till now we perform only lab experiments, so the other optimization methods are remaining.
So just quickly, what is the normal range of that enzyme that you want to monitor in serum?
Sir, okay, in healthy serum the sorry in healthy serum the normal range is around is 40 to 160 unit per liter. So if we could detect this limit, it's good enough. And there are higher other than means here the purpose is that alkaline phosphatase is increasing this disease like liver disease, bone disorders. So that we can easily detect since the LOD is 936 unit sorry micro units per ml.
Yeah, and but that is under the liquid reaction, right? You did it in a liquid medium.
Yes, sir. It is in
Right?
serum sample.
Yeah,
Yes.
that cannot be compared with what you are proposing. The solution you are
Yes.
proposing is a paper base. The reaction will happen on the solid medium, right?
Yes.
Yeah, so that
Yes.
is altogether different exercise you have to do. It has nothing to do with what you have to run here 90 micro units and all. Right, so that's a
Yes.
different ball game. And you in the liquid medium, you have shown the stability for not even 90 microseconds. I mean nanoseconds for that matter, right? Lifetime study what you have shown.
Yes. Yes, sir.
Yeah. So when you are going
It's
for color comparison, the stability of the color produced is important. How long it stays, right? The reaction time and everything is important. I think it is very primitive data right now, or it's more like a you're trying to build a concept. Is what I figure out from the data that you have
uh,
shown.
yes. Actually, it is till now in very few months we propose we developed with our lab base. Now it's just it was just initial phase that we are moving for paper base. So
Yeah.
the remaining time to
Yeah.
develop. Yes.
So it's important when you think of such solution, first see what are the disadvantages of available solutions. If available tests are more accurate, specific, quantitative, then your test will not have any standing, right? It will not be helpful. So you need to first put that comparison, like what is the problem? Like the levels increase, that is one thing, but already there are solutions to measure those levels accurately. So what is the disadvantage of those? That's what exactly Madam was also asking. What are the drawbacks of the existing solutions or existing tests which you are trying to address? So think on those lines.
Yeah.
Yeah.
Okay,
Thank
sir.
you. Thank you.
Sir, okay. Am I answer to these drawbacks of the existing system? Or,
Yeah, you didn't answer that.
oh, yes, I will give. So the drawbacks are first of all high cost, and there is this incubation time. Since some are colorimetric, some are fluorimetric, but they are not point of care device. If we are proposing and we developing the paper-based sensor, it will be a point of care device also, and it will give quick response. So in biomedical work. While working with the biomedical worker, we can see that in short period of time we need to be like we need to do lots of work, and in that it will be save their time, and it will give means the response as well as sensitivity because of the this system is ratiometric, There is no environmental factors like pH, salt will be affect this system. If it is affect, the response will be not affected. So this is a main advantage of this system. But in case of the traditional method and single fluorescence method, this is the major limitation and we can say drawback that it can be affected because of the other environmental factors, and there is more assay time requirement uh, plus high sensitivity, and there is no lack of the point of care device. This is the drawbacks I can say.
Yeah, but there is a disconnect between the problem statement and the solution you are designing, is what I could figure out. Thank you.
Sir, my problem statement is to detect the alkaline phosphates, which is means here basically here I'm not compare. We can say here I'm not comparing the commercial kit with uh, uh, the traditional methods, but here we are just trying to improve in existing methods so that the all drawbacks will be overcome.
Okay, thank you.
Thank you, sir.
Thank you, judges, for the question and answer session. And now I would request the judges to please say a few words on this session.
Okay, so students, we had 7 presentations today.
Yes,
Yes, ma'am.
All the entries were very good. I mean, we would like to hear from judges, their views.
I thought Dr. Meenal would start, but I'll take this initiative again.
Yes, so I want you to start first.
Yeah, please.
Go ahead, Minal madam.
Okay. Yes, so all presentations were definitely, it was a good attempt by all the participants. What I would suggest you all go back to your ideas and look at it from a commercial perspective. How can you use this idea and go ahead to a commercial level? Right now, there are many loopholes. I'm sure you will work on it with whatever suggestions have been given, or the question that were asked will help you to build up on your idea to a scale up level. Because it's very important that whatever idea you are working on, you should be able to bring it to the market, because that is which will give you a great success where it will reach the society. That is what I would like to say. Thank you. Thank you from my end.
Yeah, thank you, Dr. Meenal. Firstly, thank you, SVKM's BNCP, Munira ma'am, all the faculties, as well as my, I would say, co-judges, Dr. Srinivas, as well as Dr. Meenal, for this specific opportunity. I just wanted to highlight one specific pointer in these entire presentations. Traditionally, we have always been speaking about pharma, typical pharma industry actually, but these 7 presentation was kind of an eye opener, I would say, for everyone, that sky is the limit actually when you are learning pharmacy. Someone speaking about mobile app, like using tech. Someone speaking about the API processes actually in terms of the biomanufacturing process. Speaking about I would say waste management as well as something like a product development, which we had anti-tobacco smokeless product. as well as lastly the diagnostic kits answer. So this actually gives us a lot of insights that we as a field in pharmacy actually have way better exploratory opportunities as far as the startups or the entrepreneurship is concerned. And I'm pretty confident that these specific presentations would help the students build their confidence as well as help them actually have a broader vision in terms of not only in terms of development but in terms of also commercialization of these kind of products, as Dr. Meenal rightly mentioned. And I believe, as Dr. Srinivas had mentioned in his presentation, that BNCP is associated or collaborated with the incubation center that Dr. Srinivas belongs to. And I'm pretty confident moving ahead, some of these products or maybe products from next generation would be something which would definitely see the day of the light. And with this, I just congratulate everyone. The idea herein was to present, it was not a race basically, but it is something which every one of you should go through that process so that it is helpful for the entire ecosystem. Thank you.
Yeah, I think it was a good opportunity to co-judge with Vijayji and Dr. Meenal. Idea per se, I'm not sure how many of these 7 entries, like I could see there were solo presenters also and there were teams. So, and I'm not sure whether they had a mentor who was guiding them on these problem statements, or it was like the exercise done by the students themselves. If it is done by students themselves, hats off to them. At this stage, they are thinking through the idea and trying to have a workable solution. But when it comes to actually identifying the problem right way, and then trying to build solution or ideas around it, I think handholding will help them. So this is an excellent exercise to look at how differently students are thinking for a certain problem to bring a solution. This is a wonderful exercise for that. And I see potential in some of the ideas where they have actually tried to work as an entrepreneur and see the various factors. Like I was surprised to see the cost analysis. I was surprised to see the market size that they have, a couple of them have shown. And that is the way to go about if you're talking about taking the innovation to next level. So, wonderful attempt by all the 7 participants. And Dr. Munira, as Vijayji has given one clue, I'll be more than happy if some of these ideas need to be curated further, and we can do handholding for these people who really want to take it forward. So this should not end as the research project or MPharm or PhD project, but can this be translated in the real life solution? So definitely we're more than happy to work together on this. So that's a few things I wanted to put here. Thank you.
Yeah, thank you very much, all 3 judges, and thank you, Dr. Srinivas, for offering the participants of SRC IIC to further get mentoring from you. Thank you very much. Over to you, students.
Thank you, ma'am. On behalf
Can I get a 1 minute? Can I get a 1 minute?
Yes, sure, sure.
Okay, because I just spoke about the participant and their ideas. I would like to take this opportunity to thank Principal Madam and my co-judges. It is my pleasure to have sharing this platform with Vijay sir and Srinivas sir. Thank you so much. Yes, thank you.
Thank you, ma'am. On behalf of the organizing team, it is my privilege to deliver the vote of thanks on this special occasion. To begin, We express our deepest gratitude and respect to our esteemed judges. Your thoughtful insights, valuable time, and profound expertise have been a guiding force for this event. Your contributions have elevated the experience for everyone involved, and we sincerely thank you for your support. We would now like to present a certificate of appreciation as a token of gratitude.
A heartfelt thank you to all the participants for bringing your energy, creativity, and hard work to this platform. You are the true driving force behind this event, and your dedication has made it truly exceptional. We are proud of each of you for your remarkable efforts. Now, I would like to conclude this session with the certificates of our judges, Mr. Shivpujay, Dr. Savle, and Dr. Rangan. Thank you.
We would be happy if our judges stay back for the valedictory.
Yes, ma'am. I would also like to add to that point. All the judges and the participants kindly move to the room 1 for the valedictory session. The link is
available in the chat
box. Yes.
Thank you.
Dr. Manira, you will have to excuse me. I have to rush for another engagement.
No problem. Thank you very much.
I
You have
wish
spent
all the best. Yeah.
You have spent the whole day with us, and I understand you have a lot of other activities to be done. Thank you very much. Really appreciate your time and effort. Thank you.
Thank you, the hosts. They have been doing commendable job.
I must tell you, they are first year student, just admitted 21 days ago.
Well done. Well done. One long way to go.
Yeah, big round of applause for them.
Thank you, sir. Thank you, ma'am.
Yeah, let's meet in the room 1. We can move to room 1.
Yeah. Bye.
Bye.